A 2023 study published in the European Journal of Radiology by Laszlo Tabar and his colleagues analyzes a specific tension within current breast cancer diagnostics. Under the current WHO classification, invasive lobular carcinoma is defined by its cell morphology, specifically discohesive cells that appear in a single-file linear pattern. In a review of 560 patients with this exact diagnosis, researchers identified two separate disease behaviors that are currently grouped together under this single definition. The long-term data for these patients reveals a split in outcomes. One group shows a 56% survival rate, while the other remains at 84%. When one diagnostic term covers populations with a 28% gap in survival, the accuracy of the underlying classification becomes a focal point for investigation. The research suggests that the current classification may be an anatomical mischaracterization of how these tumors actually grow. To differentiate these outcomes, the study utilized a combination of mammographic imaging biomarkers and large-format thin-section histopathology. This approach allows for a new framework based on the specific site of tumor origin within the breast tissue. Moving towards precision oncology requires transitioning from these broad diagnostic categories to definitions that reflect the anatomical site where the malignancy begins. We begin with the first subtype, which is characterized by its presence in the extralobular mesenchyme. On a mammogram, this variant presents a very specific set of imaging biomarkers. There is extensive architectural distortion throughout the tissue, but no visible central tumor mass. Correlating this with large-format histopathology confirms that the malignancy is not a solid lump, but a diffuse infiltration. Within this infiltration, we see the classic pattern of discohesive cells in single-file lines. This diffuse anatomical presentation is the one associated with the 56% long-term survival cohort. Because this growth occurs in the extralobular space, it spreads unconfined through the mesenchyme, leading to a more extensive disease presentation. The second variant originates entirely within the intralobular mesenchyme. The mammographic biomarkers for this group offer a sharp contrast. Instead of diffuse distortion, we see a distinct unifocal tumor mass, typically under 20 millimeters. Histopathology confirms these cells aren't spreading diffusely, but forming a localized colony. The malignancy is strictly confined within the terminal ductal lobular units, or TDLUs. This contained unifocal presentation is found in the cohort with 84% long-term survival. By remaining within the structure of the TDLU, this subtype presents a different clinical profile than the unconfined extralobular variant. These findings challenge the term lobular, which implies that the cancer originates from the luminal epithelial cells that line the lobules. If we examine the individual acini within the TDLU, the cellular evidence becomes clear. The luminal epithelial cells lining these structures are normal and remain entirely intact. We prove these cells have different origins using E-cadherin staining. The normal epithelial cells of the acini stain a strong brown, indicating E-cadherin positivity, but the surrounding invasive cells are uniformly negative. This complete loss distinguishes the malignancy from normal epithelial cells. This suggests the cancer is not epithelial in origin, but arises through a process called mesenchymal epithelial transition, or MET. In this model, hybrid cells within the mesenchyme undergo a transition into the invasive cells seen in these cancers. The location of these cells, either in the extralobular or intralobular space, dictates which disease subtype develops. Calling this lobular carcinoma is an anatomical misnomer that attributes an epithelial origin to a disease that actually begins in the mesenchyme. We are looking at two distinct diseases currently unified by a single name. On one side, we have diffuse architectural distortion. On the other, a unifocal mass. These structural differences result in a 28% gap in survival outcomes. Treating a localized disease with the same intensity required for diffuse, unconfined infiltration can lead to significant variations in patient management. The localized intralobular variant is primarily a surgical disease, and in many cases, patients may not require postoperative radiation. The diffuse extralobular variant requires an entirely different approach because it infiltrates the tissue so extensively. Using imaging biomarkers to split this diagnosis prevents both the overtreatment of localized disease and the undertreatment of diffuse disease. Current terminology fails to distinguish these entities, but the imaging and histological data provides a clear path forward. This requires a shift in interdisciplinary communication, where radiologic imaging biomarkers are integrated directly into the pathology report. For the diffuse cohort, the study proposes the term breast cancer of extralobular mesenchymal origin, or extralobular BCMO. For the unifocal cohort, the term is breast cancer of intralobular mesenchymal origin, or intralobular BCMO. We now need clinical trials designed to optimize specific treatment protocols for these two distinct subtypes. Precision oncology demands that we name the disease according to its origin, moving past the inaccurate umbrella of classic invasive lobular carcinoma.